Have you ever wondered why you and your family look slightly, but not entirely alike? The way in which traits are passed down from generation to generation was first explained by Augustinian monk Gregor Mendel. So how did the monk become known as the father of genetics? Well, in the mid-18th century, Mendel bred over 30,000 pea plants together for their characteristic features. In one of his experiments, he crossed pure lines of purple and white flowered pea plants. Mendel noticed that the purple flower masked the other color and he called it a dominant trait, while the white flower was recessive, meaning it would always be masked unless paired with another recessive allele. Today, this is now known as one of Mendel's 3 laws of inheritance, the law of dominance. But it wasn't until the early 1900s that one of his successors, British geneticist Reginald Punnett, realized that in order to keep track of which plant combinations produce which flower colors, he needed some sort of charting tool. Thus, the birth of the Punnett Square. What is a Punnett Square and how does it work? The Punnett Square is a table that lists potential pairings of paternal and maternal alleles. These tables can be used to look at the genotypic odds of a single trait or of crossing numerous traits from the parents. Biologists use the figure to calculate the likelihood that a child will have a specific genotype. The phrases heterozygous, homozygous, double heterozygote or homozygote, dominant allele, and recessive allele are crucial to comprehend when using the Punnett Square method. Monohybrid. Each genetic locus is always represented by 2 letters and the prefix mono means 1, indicating that this cross refers to the investigation of a single feature. For example, Punnett squares with each cell having a theoretical 25% probability provide probabilities of the coloration status of their offspring for each combination of the parents. Dihybrid. Examining 2 or more genes enables the creation of crosses that are more intricate. However, for the Punnett square to function, the gene must be independent of one another, which implies that having a specific allele of gene A does not change the likelihood of having a particular allele of gene B. This is the same as saying that the genes are not connected, which prevents the 2 genes from naturally sorting into one another during meiosis.